Hi Emma, I've been reading about microscopy techniques, but I'm curious. Why do we use electron microscopy?
Excellent question. We use electron microscopy, or EM, to study structures within cells that are too small to see with light microscopes. Electron microscopes allow us to visualize cell organelles, macromolecules, viruses, and even subcellular structures in incredible detail.
How does it work? What makes it so powerful?
Electron microscopes use a stream of electrons instead of light. This stream is generated by an electron gun, which produces a beam that behaves similarly to light, but with a much shorter wavelength. Because of this short wavelength, electron microscopes have a very high resolving power, allowing us to see details on the nanometer scale. In fact, depending on the technique, EM can achieve magnifications of up to 1,000,000 times.
So, what's the difference between electron microscopes and light microscopes?
Light microscopes use glass lenses to focus light, while electron microscopes use electromagnetic lenses to manipulate the electron beam. EM images are captured in black and white, although color can be digitally added later to highlight specific structures.
I've heard there are different types of electron microscopes. What are they?
There are two main types: transmission electron microscopy, TM, and scanning electron microscopy, SEM. Each one has distinct capabilities. Let's start with TM.
Sure. What does TM do?
TEM is designed to observe internal structures within cells. In TM, electrons are transmitted through an ultra-thin specimen, usually less than 500 nanometers thick. The electron beam passes through the sample, creating a highly detailed 2D image of the internal structures.
And how are these samples prepared?
Preparing samples for TM is meticulous. The samples need to be carefully dehydrated, as water interferes with the electron beam, and they are often fixed using chemical or cryomethods to preserve structures. They're also coated with heavy metals like gold or platinum to improve contrast.
What's the magnification like with TM?
TEM can magnify up to 1,000,000 times, with a resolution down to 0.1 nanometers. It's highly effective for studying organelles, proteins, and even viral particles within cells.
What about SEM? How is it different?
SM is excellent for examining surface structures. Unlike TM, which looks inside, SEM scans the surface by bouncing electrons off the sample's exterior. This gives a 3D image of the sample surface, which is particularly helpful for looking at the textures and shapes of biological and non-biological materials.
That sounds useful for many fields.
It is. SEM is popular because sample preparation is relatively straightforward. Samples are coated with a thin metal layer, often gold, to improve electron reflection. With SEM, we get magnifications up to 1,000,000 times, with resolutions around 0.5 to 1 nanometer.
Moving on to another topic. What about vesicular transport? I read that it's essential for cell function.
Yes, vesicular transport is crucial. It's how cells move materials in and out. We can break it down into two main processes: exocytosis and endocytosis.
Could you explain each one?
Certainly. Exocytosis is how cells expel materials. For example, cells constantly produce ECM proteins, digestive enzymes, and hormones. In exocytosis, vesicles containing these materials merge with the cell membrane, releasing their contents outside the cell.
So, exocytosis is like the cell spitting out what it doesn't need?
Exactly. Then we have endocytosis, the process by which cells take in substances. There are different types: pinocytosis, or cell drinking, where the cell takes in fluids, and phagocytosis, or cell eating, where it engulfs larger particles like debris or microorganisms. Another type, receptor-mediated endocytosis, is very specific. It allows the cell to absorb targeted molecules like certain hormones or nutrients.
I've heard about transcytosis too. How does that fit in?
Transcytosis is fascinating. It's a specialized process where substances are transported across the entire cell. This happens in cells with a high rate of material exchange, like in the lining of the intestines. The substance is absorbed via endocytosis, transported through the cell, and then released on the opposite side via exocytosis.
Let's shift to the cytoskeleton. What are its components?
The cytoskeleton is the cell's structural framework. It has three main components: microtubules, microfilaments, and intermediate filaments.
I'm familiar with microtubules. They're involved in cell division, right?
Yes, microtubules are made of tubulin proteins and play critical roles in cell division and transport. They form a structure called the mitotic spindle, which helps divide chromosomes during cell division.
And microfilaments? Are they similar?
Microfilaments are thinner and more flexible. They're primarily composed of actin, which interacts with another protein called myosin. This interaction allows the cell to contract and move, making microfilaments essential for cell motility and shape.
What about intermediate filaments?
Intermediate filaments provide tensile strength, helping the cell resist mechanical stress. They're more stable than microtubules and microfilaments and form a supportive network throughout the cell.
I've also read about molecular motors. How do they work with the cytoskeleton?
Molecular motors are proteins that move along the cytoskeleton, powered by ATP. There are three main types: myosin, kinesins, and dyneins.
What's the role of each type?
Myosin move along actin filaments and are crucial for muscle contraction and transport within cells. Kinesins and dyneins travel along microtubules but move in opposite directions. Kinesins typically move toward the plus end, helping transport organelles and vesicles toward the cell's outer regions. Dyneins move toward the minus end, often carrying materials inward and playing a role in the movement of cilia and flagella.
It's incredible how much goes on inside a single cell. Thank you for explaining all of this in such detail.
You're very welcome. Electron microscopy, vesicular transport, and the cytoskeleton all reveal the cell's complexity, helping us understand everything from basic cellular structure to how diseases develop at the microscopic level.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.